Welcome everybody. My name is Suyash. I'm a current senior at Texas Tech, pursuing a major in microbiology as well as a minor in chemistry. Today, the title of the poster that I'll be presenting is Dental Sealant Containing Organo-Selenium Compound Inhibits Bacterial Biofilm Formation by Oral Bacteria. So originally this information was presented in more of a poster format, but due to the virtual nature of the conference, I took the same information and transferred it to a PowerPoint. I have linked the PDF version of this poster in the description below, so feel free to download it and follow along with me. So let's start out with an overview. The reason why we're doing this experiment is that studies have found biofilms form readily on tooth surfaces and pose major health problems such as cardiovascular disease and tooth decay. In fact, Streptococcus mutans is thought to be the primary cause of biofilm formation. So the purpose of this experiment was to determine whether selenium-containing dental sealant promotes superoxide production and can be a cheap, reliant form to prevent biofilm growth. Using colony-forming unit assays over 6 logs of inhibition, we found that just 0.2% of selenium surface sealant to be effective at preventing the growth of Streptococcus mutans. This led us to conclude that dental sealant containing organoselenium provides strong evidence of inhibiting bacterial growth in oral cavities and has a wide range of applications, including easier oral care for young children with braces. So what even are biofilms? Well, biofilms are a conglomeration of cells that grow and work together. There are 5 stages in which they form. First is attachment. This is where planktonic cells deposit on various surfaces. Second is multiplication. This is where bacterial cells undergo mitosis and they grow and divide. Third is exopolysaccharide. This is where bacterial cells secrete carbohydrate polymers, which is a sticky substance that prevents the immune system from accessing the bacterial cells, as well as prevents antibiotics from reaching and killing these bacterial cells. Fourth is maturation. This is where the biofilm continues to grow and excrete more exopolysaccharide. Finally, is succession. This is where planktonic cells are released from the now mature biofilm. It's important to note that it is at this point that the immune system can now target these new planktonic cells. But even if the immune system is successful in killing all the planktonic cells that are released, the original source of the infection, which is the biofilm, still remains. Streptococcus mutans specifically encourages biofilm formation in the mouth by interacting with other bacteria. The study found that this was primarily via quorum-sensing molecules. And from chemical isolates, we found that Streptococcus mutans is resistant to many of the traditional antibiotics that we would use to treat these sort of infections. Streptococcus mutans also produces glucans, which can cause heart problems. This is because Streptococcus mutans can enter the blood vessels that are present in the mouth and travel to the heart. This causes infective endocarditis, which is an inflammation of the heart, which can cause a whole host of health problems later down the road. So now that we've talked about biofilms, let's talk about a possible treatment. In this experiment, we focused on selenium. Selenium has high catalytic activity when covalently bound to surfaces. This leads to its first major advantage, that selenium doesn't have to leave the surface to which it's bound to act upon these biofilms. Production of superoxides requires only oxygen and thiol as reducing agents. So this means that selenium can produce these superoxides, which are oxygen radicals, which can break up the DNA of bacterial cells and cause them to die without much help. Only oxygen, which is present in the mouth, and thiol, which is present in most, if not all, bodily fluids, as well as on the surface of bacteria. The second advantage of selenium is that small concentrations of selenium are sufficient to maintain appropriate catalytic activity. So not a lot of selenium is needed to prevent this biofilm growth. Finally, selenium is also very cost effective, which is a huge advantage over its major competitors such as silver. There are 4 main steps that we did in conducting this experiment. First, we mixed commercial sealant with selenium. The commercial sealant was comprised of TEG-DMA as well as bis-GMA, and the selenium was a diacetyl methacrylate. These are all just polymers that harden under UV light. The second step was curing. The mixture was pipetted into 1.5 milliliter centrifuge caps, and the UV light was used to harden the sealant. Finally, the samples were washed with PBS for 5 minutes and dried. Finally, the samples were sterilized in an autoclave under the gravity instrument cycle. So as you can see from the graph, we used colony-forming unit assays to determine whether the selenium was successful at preventing biofilm growth. Selenium was incubated at 1 milliliter BHI and 150 microliters of glutathione and inoculated with 10 to the third colony forming unit assays. After 24 hours, we obtained 10 to the 6 CFUs from the control sealant and 0 from the 0.25% selenium surface sealant. This led us to conclude that 0.25% selenium inhibited 6 logs of Streptococcus mutans. To confirm these results, we use scanning electron microscopy. As you can see on the A or the left side, Streptococcus mutans grew in high concentrations on the untreated control, whereas on the right B side, there was no Streptococcus mutans grown on the selenium sealant. There are 3 conclusions that we obtained from this experiment. First, selenium can be mixed with commercial sealants in a stable manner. Second, organoselenium containing sealant inhibits over 6 logs of growth. And finally, small concentrations of organoselenium are sufficient to prevent biofilm growth. Now, there's a lot of future research that needs to be done before this can be commercialized. First of which is that the effect of organoselenium on other microorganisms in the mouth must be observed. This is because even though Streptococcus mutans is one of the primary biofilm-causing agents in the mouth, there's other bacteria also present in the mouth. We also need to test the effectiveness of selenium on various devices such as dentures. There are 2 main impacts to this study. First is to provide easier oral care for young children with braces. A lot of the time with braces, what we see is that the sealant or the actual instrument themselves prevents young children from brushing their teeth properly, which can cause cavities and can lead to cardiovascular disease. The second impact is to help the elderly with physical and or mental disabilities better care for their health. A lot of time, the elderly can't perform tasks such as brushing their teeth, and so using any sort of instrument to help them with this small task would greatly improve the quality of their life. At this point, I would like to acknowledge a few people. I'd like to thank the Honors College Undergraduate Research Scholars Program supported by the CH and Helen Jones Foundations. Additionally, I would also like to thank my mentors, Dr. Train and Dr. Reed at the Texas Tech Health Science Center. Here's some references that I used throughout the poster. Thank you once again for taking the time to join me on this presentation.